Hi, everyone. Now we're going to start the neuroanatomy module. So first, a little bit of history of the brain and our understanding of brain structure. The first written reference to brain was nearly 5,000 years ago in ancient Egypt, where there was a battlefield report. As a TBI researcher, this is particularly interesting to me. But these are the hieroglyphs that corresponded to brain. One thing to note, which we'll understand in the coming slides, that I think is interesting is this folded cloth hieroglyph. This is a representation of the one of the key visual features of the brain when you see it is that it has these bumps called gyri. So this is a reference to the folded gyri of the brain. So the direct translation of these hieroglyphs is something like skull offal or skull meat or skull organ, which tells you that they really didn't have a great understanding of how important the brain is to our personality, our intelligence, all of that. But they did at least recognize that it was recognize it as an organ. So in about 5000 BCE, Alcmaeon in ancient Greece was the first known to hypothesize that the brain is responsible for thought. And in the 1500s, Vesalius proposed that nerves stem from the brain and transmit sensory and motor information to and from the body. So a big step forward. Slight step back in the early 1800s with the pseudoscientific field of phrenology. This was a field where they thought that they could link bumps on the skull to character traits, such as a heavy brow being indicative of criminal behavior. Obviously, we now know that that's silly. That's not how things work. And in the mid to late 1800s, lesion and histological studies moved the field of neuroanatomy forward. With lesion studies, scientists were able to hypothesize that certain brain regions were responsible for certain functions based on what function was impacted if there was an injury to a particular brain area. And with histological studies, looking at different cell types, researchers hypothesized that regions that had different cell types might have different functions. So first, getting oriented, some of the terminology that we're going to use when we're looking at brain structures. We have superior or dorsal, inferior or ventral, anterior or rostral, posterior or caudal, and then medial or in the middle, and then lateral on the sides. So the reason that some of these have two names is one that is more applicable for quadrupeds and one that's more applicable for bipedal animals such as us. So superior and inferior makes sense to us, and anterior and posterior. But rostral caudal coming from cauda, meaning tail, that would be in a quadruped, the part of the brain that's closest to the tail. And since this is a neuroimaging course, you're going to see a lot of MRI images, and it is, in addition to being able to reference the orientation, we also need to talk about the particular plane that we're looking at, what the image is. So obviously the brain is 3D, but a lot of the images that we look at are 2D. So we need to talk about the different planes that we're looking at. These are the coronal plane, which goes across like this. We have the sagittal plane, which goes down the middle, and the axial plane, which can also be called horizontal. And here those are represented on a 3D reconstruction of my brain. So one of the key features of the brain that I referenced on that first slide that the ancient Egyptians noticed is the gyri and the sulci. A gyrus is a bump on the brain, and the sulcus is the valley in between. Gyri and sulci is the plural here. So here, for example, you can see the human brain and how bumpy it is, all of the valleys, all of the bumps. Here with the sheep brain, still some of those, but less. And then here, the rat brain is totally smooth. So we have what is called a gyrencephalic brain, same with sheep, whereas rats and other less evolved animals have what is called a lissencephalic brain, meaning that it's smooth. And this gyrencephalic pattern is critical for our development, our intelligence, our evolution. We were limited in how large our brain could get by the size of the skull. And the size of the skull is limited by the size of by the size of a woman's hips and their ability to give birth without dying in childbirth. So brains could only get so big before we weren't really going to survive. So how to pack more information in is gyration. So the gyrencephalic brain has all of these bumps and curves, and that means that it has greater surface area, so that the volume is the same, but the surface area is larger. Total volume is the same. Some gross anatomy. So every brain is a little bit different, but there are certain landmarks that aren't nearly always present, and those are represented here. So we have the central sulcus in red, which divides the frontal lobe in orange and the parietal lobe in blue. We have the lateral fissure or the Sylvian fissure here in green, which separates the frontal and parietal lobes from the temporal lobe here in yellow. Some other important regions that we'll talk about, the occipital lobe here in the back. We have the cerebellum here in green and the brainstem down here in pink. And here are some of those fissures and sulci visible on MRI data. So here, running down the middle of the brain, the longitudinal fissure, this wasn't represented in the last slide, but that's the fissure that separates the left and the right hemisphere. You can also see the central sulcus here that separates the frontal and parietal cortices. And on either side of the central sulcus, there are two gyri that take their name from this. So we have the precentral gyrus, which is in front of the central sulcus, and the postcentral gyrus, which is behind the central sulcus. You can also see here the Sylvian fissure separating the temporal lobe from frontal and parietal cortex. And now a medial view. So middle as opposed to the lateral view that we were taking before. A few more key regions become apparent. We have the parieto-occipital sulcus here in dotted red, and this separates the occipital lobe from the parietal lobe. We have the corpus callosum here in white in the middle, and this is the main connection between the left and the right hemispheres. So the cerebral cortex is divided by the longitudinal fissure. The corpus callosum is the bridge between the two hemispheres so that they can communicate. We have the thalamus here. This is a really key relay station for body and brain to communicate. We have the hypothalamus. Hypo just means less or under, so under the thalamus. And the hypothalamus is important for generating and circulating hormones. And these are critical for all aspects of our lives, for growth, for reproduction. And here we have the cingulate sulcus in blue, which separates the cingulate gyrus, which is underneath it, from the frontal and parietal lobes above it. And here in these GIFs, you can see the limbic lobe. Some people don't call it a lobe, limbic system, whatever you call it. These regions are related to each other in green. We have the cingulate gyrus, which is this region that you can see running over the corpus callosum, running on either side from the front to the back. And then we have the hippocampus and the amygdala, more on the lateral edges that are connected. You can see them there. And the last lobe is the insula, the insular lobe. Insula means island, and it really is separated from the other ones. It's underneath all of the other ones. You can only see it if you carve away the frontal, parietal, and temporal lobes. And this is another good representation of how the brain really increased its surface area and its capabilities without having to increase size. It actually folded lobes on top of each other. So some functions of the cortical lobes. Now there are some, take this as a general guide. There are very few things that are only located in one part of the brain. If you could think about, a system that you want to be robust, that would not be a good idea if you only had one part of the brain that was important for each each function. If that part of the brain gets damaged, what are you going to do? So there's a lot of overlap, but there are some general guides that you can take in terms of what brain regions do. So very generally, the frontal cortex is responsible for executive functions, decision-making, planning, personality. A lot of those, what we call higher order functions that you see that we think define us as humans as compared to other less evolved animals, like dogs, for example, can't really plan many days ahead. We have in the parietal lobe, primary somatosensory cortex, which is the part of the brain that's responsible for receiving sensory inputs, and association cortex, which is important for integrating these sensory inputs and helping us to interpret them. We have the occipital lobe in back. This is the primary visual cortex. So obviously our eyes see things. This information is transmitted back to the occipital cortex, which is where it is processed and moves forward so that we can interpret what it is that we're seeing. And the temporal cortex here. The temporal lobe contains primary auditory cortex and is also responsible for language comprehension. So this is both hearing and speaking. The limbic lobe, again, which is here in the center, is important for memory and emotion. And this is one of those kind of fuzzy functions that pulls from a lot of different brain regions. So there isn't one part of the brain that's responsible for emotion or one part of the brain that's responsible for memory, because these are really complicated functions. But the limbic lobe, one of its important functions is supporting memory and emotion. And then lastly, the insular lobe, which is important for pain and visceral sensation and helps integrate taste and smell. So now moving on to different tissue types of the brain. These are divided into gray matter and white matter, which really just comes from how they look. Whether you are looking at them on an MRI like this one right here, or you're actually looking at it in person, the gray matter appears more gray and the white matter appears more white. So the gray matter, which is here in red, not that gray, obviously, it's highlighted in red, is what we also call the cortex. Cortex means bark. It's like the outside layer. And the gray matter is made up of the neuronal cell bodies called soma. So here is a neuron. The cell body, or the soma, is here, where all of the processing occurs. The nucleus is there. And these dendrites are responsible for going out and making other connections. We're going to talk about the axon and such in the next slide. So all of the processing occurs here in the soma. So if you have seen a picture of results from a functional MRI study where they have individuals look at a scene or read a passage, and you see parts of the brain light up, that's gray matter that generally lights up. fMRI researchers don't say when you say they don't like the term light up, but that's kind of what it looks like when you're first learning to look at the data. And white matter, which is here highlighted in blue in the center of the brain. White matter is white because of the myelin. So here is the axon, and you can see these little white bundles along the axon. These are parts of the myelin sheath. So each of these is a cell, Schwann cell, which is made of fat, and it encases the axon and insulates it and allows for greater transmission, faster transmission, more efficient transmission of information. And this is exactly like insulation in electrical wiring. If you have wiring that doesn't have insulation, you're going to lose signal along the way. So fibers that are well insulated, they aren't going to lose their signal. They'll be able to transmit information more quickly. And here, taking a look at what white matter looks like in a dissection. So in this view right here, this coronal section of the brain, if you were to cut along this blue line right here, cutting away the parietal cortex, the temporal cortex, the insular cortex, you would see something like this with all of these fibers rising up and radiating out. These are the white matter fibers that connect the cortex to the rest of the body. So here's another way that we can represent white matter. On that last page, that was pictures of a dissection. And this is a reconstruction of white matter. So this is using a particular type of MRI called a diffusion MRI, which we'll talk about in some upcoming lectures, which is sensitive to the movement of water and allows us to reconstruct and model the brain, the white matter tracts. So here is an example. In this, you can imagine the person looking this direction. That's the view that we have. And the colors correspond to the direction of the main orientation of the connection. So red is fibers that go from left to right. So mainly the corpus callosum is what we see in red. The blue fibers are the ones that go from superior to inferior. So just like that last slide where you saw the radiations, those are a lot of the fibers that you're seeing here that would be in blue in this representation. And in green are the fibers that run anterior to posterior. So we can classify white matter tracts into 3 categories based on what regions they connect. One classification is projection fibers. These are fibers that connect the cortex to other parts of the central nervous system, such as going down the brainstem and the spinal tract. And this is mainly represented in the corticospinal tract. We have commissural fibers. These are fibers that connect the left and the right hemisphere. The main commissural fiber system that we think about is the corpus callosum. And we have association fibers, which are basically everything else, everything that connects cortex to cortex within the same hemisphere. So for example, some of these green fibers are the uncinate fasciculus, which is a brain region that connects the medial prefrontal cortex to the amygdala.